Hello everyone and once again welcome to BioLine. Today's video is another video in the lecture series of microbiology. Okay, today we will be talking about a very common staining technique which is known as the gram staining technique. Okay, this is based upon the differential component of the bacteria in the cell wall. Okay, I have already talked about the cell wall, about the peptidoglycan layer, okay, the component of those peptidoglycan layer and the differences between a gram negative and a gram positive cell bacteria positive bacteria's cell wall in my previous video. So all of you please watch that video first, then come and watch the staining technique video. It will become more easy for everyone to understand. Okay, I'll also mention the link of that video in the description box. Okay, so please watch that video and then come and watch this one. Okay, so let us start today's video. So, first of all, I'll just give you some introductory part of gram staining technique, that it was discovered by a Danish scientist who was also a physician known as Hans Christian Gram in 1884. Okay, so to honor him or on the basis of his name only, that staining technique is known as the gram staining technique. Right? Remember that this is an example of a differential staining technique. Now, what do you mean by differential staining technique? The technique which is going to differentiate between two categories of the organism. So here this staining technique will discriminate between the two group of bacteria, and one will be known as the gram positive bacteria and the another one is the gram negative bacteria. Okay, now this differences is on the basis of the ability of the bacteria to retain the specific color of the stain that we will be using in the gram stains. Okay, now, and this technique is actually very, we can say, the initial step, or we can say most widely used technique in microbiology field. Why? Because it is used for the initial characterization and classification of the bacteria. Okay, we'll be able to understand the component of the cell wall, what kind of cell wall is present in the bacteria, and on the basis of that we can categorize the bacteria. Okay, so we'll move further. First of all, we'll just have a gist of the staining procedure. Okay, so first of all, what we have to do in the very first step, we have to heat fix the bacteria on a slide. Fine, we can directly take the bacteria on the slide and just warm it and little bit heat fix it so that the bacteria may get attached to the slide surface. Fine, then we will be applying our primary stain, that is the crystal violet stain. Our crystal violet stain will be the primary stain and it will give the a purplish or a bluish color appearance to both the type of the bacteria. Okay, after that we will apply the mordant, that is the iodine. Iodine here is going to act like a mordant, right? The iodine we are going to add here. Then we will go for a decolorization step. Okay, and in the decolorization step we'll see the differences between the two categories of the bacteria. The gram positive will still remain purple and gram negative will become colorless after the decolorization step. Okay, lastly we will be adding our counter strain, that is safranin, and the gram negative bacteria which was colorless in the previous step will now gain that color of the safranin and appear pinkish, or we can say reddish in color. Okay, so these are the staining step, or we can say the basic procedure, the protocol of the staining techniques. Fine. So all these dyes we have to mostly keep around for 1 minute. That will give us a better result. But for the decolorization step, we have to only keep the alcohol for 5 to 10 seconds. Okay, we'll talk about that why it is only that we have to keep the alcohol only for the this much short duration. Okay, so this is the general procedure. After that we'll try to understand the principle, but before that we just need to understand the differences in the gram negative and the gram positive bacteria cell wall. You can see here, this is the gram positive cell wall and this is the gram negative cell wall, right? In the gram positive you can very clearly see this is the plasma membrane, and it is outer to this plasma membrane there is just only a single layer made up of peptidoglycan, but this peptidoglycan is quite thick, is approximately 80 nanometer thick is the peptidoglycan layer, and this peptidoglycan layer alone constitute the cell wall in the case of the gram positive bacteria. Okay, apart from that, if we talk about gram-negative bacteria, so we can see that there is a plasma membrane, the pink color one. Then we have a peptidoglycan layer, and outer to that we have another layer known as outer membrane layer. So in the gram-negative bacteria, we can see the cell wall consists of the outer membrane as well as the peptidoglycan layer. Okay, but you can see here the peptidoglycan layer is very thin. Okay, it is around 5 to 10 nanometer thin. Okay, just 5 to 10 nanometers the thickness of this peptidoglycan layer which is present in the gram negative cell wall. Okay, so there is a huge difference between these two bacteria. In the gram positive only one layer but thick peptidoglycan, but in the gram negative we can see two layers are present in the cell wall, the peptidoglycan, a thin layer, and then a outer membrane is also present in the gram negative cell wall. Okay, if we just elaborate the structure of the cell wall, this is how it looks like. This is the gram positive bacteria. As you can see, a thick layer of peptidoglycan is present here. Okay, then a very minute or even sometime even this is absent, the space between the peptidoglycan and the plasma membrane, which is known as periplasmic space. Okay, mostly this is very rarely visible in the case of the gram positive bacteria. Okay, also you can see in the peptidoglycan there are some acidic component, that is the teichoic acid. Okay, these teichoic acid can directly join to the peptidoglycan or they can join to the plasma membrane's lipid, and at that time they are known as lipoteichoic acid. Okay, this is the cell wall of the gram positive bacteria. Now, if we'll see the gram negative bacteria, you can see it is little bit more complex than the gram positive bacteria. Okay, you can see here this is the plasma membrane, and just upper to that there is a peptidoglycan layer, and this is a very thin peptidoglycan layer, only 5 to 10 nanometer thickness. Right, apart from that, in the outer towards the exterior side of the peptidoglycan, you can see there is the outer membrane layer, which is almost, we can see, which is also a bilipid layer as the plasma membrane is there. Right, and you can see in the outer membrane there are these long structures, okay, which are known as lipopolysaccharides, LPS, very important one. These are only present in gram negative bacteria and absent in the case of the gram positive bacteria. Right, this lipopolysaccharides, you can see, consist of the O antigen, which is just projecting outward. It also has a core A molecule and the lipid A molecules. Okay, I have already discussed about this lipopolysaccharide in detail in my previous video. Okay, so right now I'm not taking it again. Fine, so this is the structural differences between the gram positive and a gram negative bacteria. Okay, now we'll understand the principle of the gram staining technique. So, in the C, the basic principle lies in the fact that in the case of the gram-positive bacteria, just now we saw the lipid content is very less. Okay, only 1 to 2% lipid content is there, and there is a thick peptidoglycan. I'll write it in the short form, that is PG, a thick peptidoglycan in the case of the gram-positive bacteria. Whereas in the case of the gram-negative bacteria, the lipid content is very high because of the presence of a huge amount of lipopolysaccharide, plus the peptidoglycan layer is very thin in the case of the gram-negative bacteria. Okay, we have to keep these key points in our mind, then we can understand the principle more clearly. Okay, so in the very first step after heat fixing the bacteria on the slide, we are just adding our primary stain, which is the crystal violet here. Okay, once we add the crystal violet stain, what happens? It will penetrate through the cell wall and the cell membrane of both the gram positive and the gram negative bacteria. Okay, in both the cases, it will penetrate through the cell wall and the and reaches to the cell membrane. Okay, and also the peptidoglycan layer in both the cases. Right, now the cell or the crystal violet, which can be written as CV, this will interact with the bacterial component and stain the bacterial cell as purple. So after addition of the primary stain, both the categories of the bacteria will appear purple in color. Okay, because the crystal violet molecule will enter in both the type of the cell. Okay, we can see here, you can see this is the gram negative bacteria, this is the lipopolysaccharide outer membrane, a very thin peptidoglycan, and the plasma membrane, that is the cytoplasmic membrane. In the case of the gram positive, you can see this is again the cytoplasmic membrane, but you can see a thick peptidoglycan layer is here. Okay, now you can see this blue color, these dots shows the crystal violet, even here and even here also these are the crystal violet. So after the addition of crystal violet stain, you can see the stain molecules will cross the outer membrane and the peptidoglycan. Okay, and this is how they're going to stain the two bacterial cell in the purple color. Okay, both of them will be purple. You can see the gram positive bacteria, the crystal violet stain, it has been trapped between the multiple layers of the peptidoglycan also, right? So this is just after the addition of the primary stain. Now after the addition of the primary stain, the next stain that we are going to add is the grams iodine. Okay, remember the gram iodine will work here as a mordant, which is actually a trapping agent. Okay, so what exactly is a mordant? Mordant is a substance which will increase the affinity of the cell wall for a particular stain. Okay, so that the primary stain may more efficiently bind to the cell wall, that efficiency will be increased by the mordant. Okay, and here the primary stain is the crystal violet and mordant is the gram positive. Also this mordant, what it will do, it will form an insoluble complex with the primary stain so that the primary stain will get trapped within the cell wall. Okay, so in this case the grams iodine will form an insoluble complex with the crystal violet, the primary stain, that is the CVI complex. Okay, and when this complex will be formed, now it will increase, we can say it is just increasing the size of the molecules of crystal violet. Why? Because it is just getting attached to it, and as a result these molecules will now get more trapped between the peptidoglycan layer or between the cell wall of the bacteria. Okay, and still after the addition of mordant also, because the crystal violet is now more strongly attached to the cell wall, both the categories of the bacteria, the gram negative and the positive, will appear purple even after by the addition of the mordant. Okay, now in the next, you can see here in the diagram, this blue one is the crystal violet stain and this black one is the mordant iodine here. You can see how this iodine, which is positively charged, and the crystal violet stain, which is negatively charged, they are interacting with each other. You can see in both the cases, as a result, what is happening? The size of the crystal violet molecule got increased, it got larger, and now it is more efficiently fixed or trapped within the cell wall in both the cases, right? And still the bacteria is purple in color. Okay, after, okay, the next step is a very important and a very critical step in the gram staining, which is known as the decolorization. Okay, in decolorization we'll be using 95% ethyl alcohol. Okay, and this serves two functions. Number one, it will dissolve all the lipids, it will dissolve the lipids of the membrane, plus it will cause the dehydration of the membrane. That means it is going to remove the water from the membrane. Okay, so this is the two important function that this particular decolorizing agent will perform here. Okay, so see here, as we know that gram negative bacteria has a higher lipid content. So what will happen when we will add the decolorizing agent, that is the ethyl alcohol, the gram negative bacteria's lipid will get dissolved. As a result, their peptidoglycan, which is very thin layer, is very much now exposed to the this decolorizing agent. Okay, and then when it will cause the dehydration, even that thin layer will get broken up. Okay, even that thin peptidoglycan layer will be broken up in the case of the gram negative bacteria, and as a result the porosity of the cell wall increases a lot. Okay, I'll just repeat it again. What happened once we add the ethyl alcohol in the case of the gram negative bacteria? You can see here, in the case of the gram negative bacteria, we have a very thin peptidoglycan layer and we have the outer membrane. So the lipid part of the outer membrane, that is the LPS, and all will be dissolved by this ethyl alcohol, and once it is dissolved, what will happen? The peptidoglycan layer is now exposed, and once it is exposed, because it is very thin layer, due to the dehydration, even this will get broken up, or even this will get, what we can say, this will reduce in size, and there will be pore creation within the peptidoglycan layer. Okay, and as a result, what will happen? The crystal violet molecule, the complex of crystal violet and iodine, that will now become free to leave the cell in the case of the gram negative bacteria. Okay, the crystal violet or I complex is then washed away from the thin peptidoglycan layer, leaving the gram negative bacteria to be colorless. Okay, once the outer membrane lipid is gone, the peptidoglycan is being lysed and pores are created. The trapped crystal violet will be released out of the membrane. Okay, now this happens in the case of the gram negative bacteria. Let us see what happens in the case of gram positive. Because the lipid content of the gram positive bacteria is very much low. Okay, as a result, what happened? This function means the alcohol cannot find any lipid to dissolve with it. Okay, also what is happening, because it has a thick peptidoglycan layer. Okay, thick peptidoglycan layer, the dehydration, when the dehydration will take place, this whole peptidoglycan layer will get more compact. Okay, it will get more sandwiched, and as a result, the central, the CV and I complex, what will happen? That will not be free to leave the cell. Okay, it will get more trapped within the peptidoglycan layers. Okay, we can say in generally the pore size of the membrane in the case of gram positive will not increase, and as a result, the CV and I complex will retain in their cell wall, and even after decolorization, the gram positive bacteria will remain purple. Okay, this decolorizing step is very critical. The reason is why I have already told you in the previous slide that we should only apply this decolorizing agent for only 5 to 10 seconds. Why? Because if we will apply this decolorizing agent for more time, it will start breaking the peptidoglycan layer of even the gram positive bacteria. Okay, and as a result, the CV complex of the gram positive will also start moving out of the cell wall. Okay, and even the gram positive bacteria will appear colorless. So our result will become a false result. Fine. Okay, so and it is a very crucial step, and if you will just decrease the time, for example, you will only keep for 1 to 2 seconds, then what will happen? This is called under decolorization. The gram negative bacteria, okay, in the case of the gram negative bacteria, during the under decolorization, okay, when the sufficient time is not given, at that time what happen? These gram negative bacteria, okay, the pores will not be created, the all the lipid will not be dissolved, and as a result, even the gram negative bacteria will remain purple in that scenario. Right, that's why this decolorization is very important in the case of gram staining. Okay, understand this with the help of the diagram here. You can see here, when we add the ethyl alcohol, after the addition of ethyl alcohol, which is a decolorizing agent, you can see the molecules of the crystal violet, because these LPS, the lipid proteins of the outer membrane, will be dissolved. As a result, this will be exposed, so the pores will be created here. Okay, and as a result, the CV and I complex has already left the cell. Okay, but in the case of the gram positive bacteria, because it is a thick peptidoglycan layer and a low lipid content, you can see the after the dehydration, these layers are getting more compact, and as a result, these CV and I complexes, they will get further trapped, and this gram positive will retain its purple color, but the gram negative will become colorless. Okay, after the decolorizing step. After this, we'll be having our final step, that is the addition of the counter strain, which is a positively charged safranin dye. Okay, this is a counter stain. This is pinkish in color, and when we will add this dye, because the gram negative bacteria, okay, because the gram negative bacteria has already become colorless, the CV and I complex has left the cell wall. Now it can easily take the molecules of safranin, and it will also appear pinkish in color. Okay, but in the case of the gram positive bacteria, because the CV and I complex is still there, the pinkish color of the safranin dye will be masked by this purple color, or we can say the bacteria will not take up the color of the safranin dye and it will still remain purple. So after addition of the counter strain, gram negative will become pinkish and gram positive will still remain purple. You can understand here. See, after the addition of the safranin, which is the counter strain, you can see this is the reddish color of the safranin, this gram negative, because it has already become the colorless one. It is free to take up this color and this will now appear reddish or pinkish in color. Okay, but you can see gram positive because still it has the CV and I complexes trapped within its peptidoglycan. It will not take up this color and it will remain purple. Okay, so this is how the staining procedure is done. This is the basic principle of the gram staining procedure. Okay, we'll just summarize the step here. In the very first step, what we are doing, we are just heat fixing the cells on the slide. After that, we are adding our primary stain, that is the crystal violet stain, and after that the state of the bacteria is purple. Okay, obviously after adding this we have to do a rinsing or a washing step here. Why washing step? To remove the excess unbound crystal violet stain from the dye. Okay, after that we will add iodine, which is going to act as a mordant and it will help the crystal violet stain to get further trapped in the cell wall, and even after that the cell will remain, both the cells will remain purple in color. We have to give a washing step even after the addition of the mordant. Okay, we have to keep these mordant and primary dye for 1 minute. Even mordant will keep for 1 minute, and after that we will wash off the mordant. Then we will be adding the alcohol, which is the decolorizing agent, and I have already told you the time should be only 5 to 10 seconds. Okay, under this condition the gram negative bacteria will become colorless, which you can see here, and the gram positive bacteria will remain purple. This is the state of the bacteria. Okay, why? We have just seen the reason. Okay, again we have to do a quick washing here so that the excess of the alcohol should move out. Okay, and over decolorization should not take place. Okay, lastly we will add up the counter strain, that is safranin. Okay, and the gram negative bacteria will take up these color and appear pinkish in color. Okay, or reddish or pinkish in color, and the gram positive bacteria will still remain purple. Okay, and again we have to wash it, we have to dry the slide, and then we can observe it under the microscope, and when you will observe it under the microscope you get somewhat this kind of result. Okay, if you're using a mixed culture of a gram negative and a positive bacteria, you can find this kind of result. Okay, see, this is a differential stain. You can see the bluish color stain. These are the gram positive stain, and in this sample, this is the sample of staphylococci. Okay, Staphylococcus aureus is the bacteria which is used here, and which is this very small circular structure. You can see in the form of grape-like bunches. You can see the bluish color, that is the gram positive Staphylococcus aureus, and this red one you can see, these are the gram negative bacteria, and in this case the gram negative bacteria which is being shown in the figure is the E. coli, which are the small rods, the small bacilli. Fine everyone, so this is how it will look like, and this is how the gram staining procedure is done and the principle is done. Okay, so if you like the video, please hit the like button, do subscribe the channel and share the video with all your friends so that more and more people can be benefited out of this. Okay, we'll be meeting in the next lecture of the microbiology. Okay, till then have a good day everyone.